my name is Sophie Baldwin, and this is the essay for protolytic activity using azocasein. Just a little bit of a background. An enzyme is a type of protein that catalyzes reactions. Enzymes lower the activation energy of a reaction in order to speed it up. For living organisms, this plays a major role in speeding up the reactions that happen in daily life, such as metabolism reactions. It also allows for the regulation of different reaction rates. The enzyme that was used in this experiment was trypsin. Trypsin is an enzyme that is found in bacteria and the small intestine of enzymes, and it helps with the digestion of proteins that were eaten by the animal. The reaction catalyzed by this particular enzyme is protein hydrolysis. It cleaves after the proteins ARG and TYR. The difference between bacterial trypsin and animal trypsin is that bacteria secretes it, and animals, it's located in the small intestine. Okay, so our question that we had was that at what pH does the enzyme trypsin have the highest activity? Yeah. And then our hypothesis was when the bacterial enzyme trypsin is placed in 8 to 9 pH, then the enzyme activity will be at its optimum activity. Okay. Our rationale was because trypsin originates in humans and living animals' small intestines, which tend to have a basic pH of around 8 to 9. And then enzyme activity will likely be at its highest at these levels of pH. So what we did was we took our experimental variable of our pH, and our conditions were that we had 3 replicates for each pH 5 through 10. So we had like 3 5s, 3 6s, 3 yeah. Okay. Our control variables were a salt concentration of 0.15 moles, a temperature of 37 degrees Celsius, and then our negative control was that we left out trypsin and added 50 microliters of the reaction buffer to the solution to maintain its volume, and then 1 negative control per pH. Then, in the centrifuge tubes, we added 450 microliters of 0.15 moles of our salt concentration reaction buffer, 500 microliters of the substrate azocasein, and then 50 microliters of the enzyme trypsin. After adding the enzyme, we incubated the tubes in a water bath of 37 degrees Celsius for 20 minutes. After we added the 500 microliters of TSA to stop the reaction from continuing and precipitate the undigested substrate, then we centrifuged the tubes at 12,000 for 2 minutes, and again at 14,000 RPM for 2 minutes to separate the undigested substrate from the supernatant. Next, we observed the absorbance of each tube by setting the spectrophotometer at 440 nanometers. The substrate we used was azocasein, and that is a milk protein. And the random addition of a sulfonamide within the substrate has a positive correlation to the absorbance measurement at 440 nanometers. Our results. The average absorbance values at, these were our average absorbance values at 440 nanometers. And so it decreased from 5 to 7, and then we got an optimum pH between 8 and 9 at 0.32 and 0.302. And our standard deviation was less than 0.01, which means that our variability was low, very low. It was very good. So here is a visual representation of that on the chart. There's our pH 8. Also, our t-test was very good. And pH 8. And then our descriptive statistics were that we had 3 replicates and were performed for each pH. Our p-value was less than 0.05, and our observance values were, as the pH increased, the color of the solution went from a lighter color to a darker color. It was like an orange-red with the enzyme, and then a clear yellowish after it had been centrifuged. So then in our discussion, our experimental design, we said that our design was adequate to test the hypothesis. the enzyme activity was measured in absorbance by the spectrophotometer. Our conclusion was that the hypothesis was supported by the results. From the pH of 5 to the pH of 8, the absorbance increased and was highest at 8 at the absorbance of 448 millimeters. The absorbance that then decreased at this pH and continued to increase, then proving enzyme activity was highest at the basic pH of 8. Changes to this experiment that we would do in the future if we had to is that the experiment would be performed again. We would perform the 3 negative controls for each pH instead of just 1. In other groups, it is debatable whether or not our results got us the best enzyme activity as the other groups did. As the other groups that did pH got pH 9 for the optimal pH. Our hypothesis stated that the pHs 8 through 9 were optimal so that their data does support our hypothesis. However, another group got the results that the higher pH was optimal, such as 10 to 11. This did not support